welcome to this month's edition of the International Cardiomyopathy Network webinar series. My name is Perry Elliott. I'm Professor of Cardiology at University College in London and St Bartholomew's Hospital. And it's a great privilege this evening to have, I suppose, two guests really. A guest chairman, John Martin from my institution, University College, and Federica Marelli-Berg from Queen Mary. John, I know you're going to do the introduction for Federica, but maybe a brief introduction for you. John, I've come to understand and learn a great deal about your qualities, but many of you will know that John Martin is a great scientist and clinician, a pioneer in platelet biology. I think, John, you were the one who I think discovered the mechanism of formation of platelets, I think, from the fragmentation of megakaryocytes in the lung. John, I mean, you have many accolades, including the ESC gold medal, but I think probably I've known you increasingly as a philosopher, a poet, an author of short stories, also an entrepreneur. And I suppose the link with our speaker this evening is that you're someone who has no barriers with regard to intellectual inquiry. And John, you've been working on trying to accelerate clinical trial development. And in that process, I think you've encountered Federica and I think had a very productive and interesting collaboration. So John, it's a great pleasure to have you here as the chairman. I'll hand over to you to introduce our speaker for this evening.
Thank you very much indeed, Barry. So it's a pleasure to introduce Professor Marelli-Berg, Federica Marelli-Berg. I met Federica several years ago, and my impression in the beginning, my impression today is she is an extraordinary woman. She studied medicine in Milan, and after that went to Pavia to do hematology, eventually coming to Imperial College, where she did a PhD under Robert Lechler. And I think she tells me that Robert Lechler had a great influence on her life, particularly in relation to immunology. She went to Barts as the British Heart Foundation chair of cardiovascular immunology. And BHF chairs are very, very difficult to get, very prestigious. And therefore, she must be very pleased to have had that. She met our other chairman today, Perry Elliott, in 2012. So that's the introduction to this extraordinary Italian woman. But just a word about IDEA. The progress of science is not primarily to do with technique, but to do with creative idea of quality. And we have to ask, where does creative idea come from? And I think it comes from an atmosphere of freedom to be creative, but also a mixture of differences within the individual and within the environment. And within Federica, we have many different influences, which I think have mingled richly, not to produce one strand, but several strands coming together in discovery in immunology. In particular, I see her as producing an Italian renaissance, certainly in my science and my relationship with her. So I think, Perry, it's a great privilege to have a lecture here today from Federica Marelli-Berg, who, as I said earlier, is an extraordinary Italian woman who brings with her the spirit of Renaissance. Federica.
So you can see my screen, I guess. And yes, so what can I say? I'm speechless. I don't know whether I can go on and give the talk now after your introduction, John. It's going to be very difficult. I can only say thank you, and that you've been as much as an inspiration to the work that we're going to do in the future, and with Perry and Alex and all that. So thank you all for this amazing introduction. So what I'm going to talk to you is my favorite field, which is immunology, but this time is a little bit adapted to cardiology, because I'm sure you're all aware that there is a big talking about this new discipline within cardiology and within immunology, which is called cardioimmunology. And I had the privilege of working on this field for now a few years. And so I'm going to show you how much immunology and cardiology are bound to talk to each other, not just now, but even more in the future. So I will focus on cardiac autoimmunity. And what is cardiac autoimmunity? So cardiac autoimmunity arises as many autoimmune disease as a consequence of predisposing factors, which obviously are linked to some genes that people express, and that are triggered by environmental factors, including viruses, drugs, and also arise as a mystery as primary autoimmunity. So the first primus movens of this, the Italian Renaissance is coming out now, is the myocardial damage. So the first thing is that myocardium becomes damaged by one of these triggers, and this initiates an innate immune response. So this is what the immune system does when it senses danger in the body, becomes activated, and it induces what we call adaptive immune response, which is going to be the focus of my talk. So adaptive response is the immune response that we know about in vaccination, for example, is mainly mediated by T cells and with the sister cells, the B cells that produce antibodies. Now, this can lead in a number of individuals to autoimmunity and to further tissue damage. The action of the T cells that you can see here is mediated by cytokines that they produce, particularly interleukin-17 and other cytokines, which I will mention to you, which activate fibroblasts and initiate a fibrotic process, as well as activating other cells or activating in a feedback other cells of the innate immune system like macrophages. And it ends up in chronic inflammation and loss of cardiac structure and function in dilated cardiomyopathy. So I need to, sorry for a second, I need to close the, can't do this, I don't know. No. Oh, yeah, I've done it. Sorry. I had a lot of picture on the side and I couldn't see my slides. So the disease, the condition that I'm going to look at today with you is acute and chronic myocarditis. It's a very generic definition, and I understand that cardiologists still argue about definitions of various entities which really come down to inflammation of the cardiac muscle. So if we talk about acute myocarditis, my clinician friend tell me that very often it is an issue to make a differential diagnosis between acute myocarditis and acute myocardial infarction, particularly STEMI, when the angiogram does not indicate any occlusion. The diagnosis is based on, gold standard, the endomyocardial biopsy, which is invasive, and because the disease presents in a patchy manner, can be sometimes can require more than 1 or 2 biopsies. I think the general average is 14, but I've read in a review that to have 100% accuracy, you need to do 16 biopsies. So it's very difficult to, and of course, there are potential accidents related to this invasive technique. Cardiac MRI is certainly a very good way to support a suspect, a suspicion of cardiac inflammation. However, while it might give you some information, it doesn't have, it's not sensitive enough to give you a proper assessment of the type of inflammation that is going on in the heart. The other issue with myocarditis, acute myocarditis, and I would say even chronic myocarditis, is that the treatment and the prognosis. So the treatment tends to be supportive, and there is a lot of controversy on the use of immunosuppressive drugs, even when the biopsy shows a very severe T-cell infiltrate. And the other issue is because many patients recover, the majority recovers, is to predict which of these patients will then go on to develop chronic inflammation and dilated cardiomyopathy. So in addition to this, myocarditis is known to be a major cause of sudden and unexpected death in young, infants, adolescents, and young adults, or can present out of the blue as an acute heart failure. And the general impression is that this is also an underdiagnosed condition, because many patients that might have a chronic inflammatory process in the heart, and they could be completely asymptomatic and eventually present with heart failure. So that it's not clear what the real incidence of this condition is. So big problems: diagnosis, treatment, and prognosis. There's lots to think about. Now, I want to take you to my love of basic science. And as a traveler myself, moving from Italy to England, I'm very interested in the mechanisms that regulate migration of T cells once they've become activated. I'm not going to give you a speech on anatomy of the immune system, but you should think about how T cell recirculation happens in real life, and what kind of universe the T cells need to navigate to be able to reach their destination. So not only where inflammation is present, which is fine with innate immunity, but also where their cognate antigen is located. And the way they do it is something that is called T cell homing, and it's a mechanism by which memory cells preferentially recirculate to the site of original injury. And they do this because once they become activated in the lymph nodes by antigen, pathogen invasion, and so on and so forth, depending on which side of the body they become activated, in which type of lymph node, what they drain, they start expressing what we call homeoreceptor. These homeoreceptors are adhesion molecules, integrins, as well as chemokine receptor. And here you have a very old, it's 20 years old, this publication, but still pretty much up to date. This is a map of what T cells need to localize to the respiratory system or to the intestines. And I'm sure you notice that for the organs, and I became interested in this because I was originally interested in heart transplantation. The organs that are normally transplanted, with the exception of the CNS, of course, do not have an address that we can look at. And the reason why this is so important is that if you know the address, you could block the migration to a selective area of the body without affecting the rest of the immune response. So you will have what, the holy grail for immunosuppression. So you can prevent immune responses in a specific site without affecting the rest of the immune system. And that is a major problem with conventional immunosuppression. So we had the luck, I would say, to discover how T cells, what molecules T cells use to migrate to the heart. And this is the phenotype of memory T cells, so T cells that have been activated and are able to migrate to the heart. They express this molecule, which is called c-Met. c-Met is the hepatocyte growth factor receptor, which had been implicated previously in the migration of stem cells from niches in the heart by Piero Anvers. Unfortunately, the paper had been withdrawn for whatever reason, but he was right. And also they express these two chemokine receptors, CCR4 and CXCR3, and the association of these two is very rare in T cell subpopulation. So what happens when the heart is inflamed? It releases this green thing that is a hepatocyte growth factor, and this reaches the draining, the heart draining lymph node either as a soluble molecule or brought by the antigen presenting cells that are fundamental to initiate adaptive immunity, the dendritic cells. And the dendritic cells activate naive T cells. They present antigen, but also they can bind and present hepatocyte growth factor. And this gives an extra bit of instruction to the T cells when they become memory and activated, and makes them upregulate the expression of c-Met and induces the expression of CCR4 and CXCR3. Now, for a while, we did not do very much with this finding. It was very basic science, and we were dreaming of preventing graft rejection by acting upon this pathway. But the drugs that were available in 2015 were a lot less selective than what we have now available. And I will tell you more about it later on. So the first thing we did, because we couldn't go down the route of treating patients with these blockers of c-Met, we tried to make a good use of these findings by looking at whether we could use the c-Met T cells as a marker of cardiac inflammation, particularly by measuring the presence of the c-Met subset of memory cells in the blood. So the idea was, with a blood test, we could try and see whether we can have a sign of cardiac inflammation. So the answer to this question is yes. And you can see here for the non-immunologists, this is a flow cytometry dot plot. We used 4 types of patients. We use acute myocarditis patient at the time of presentation, and this is not supported by endomyocardial biopsies because they're not done, at least at the moment, at Barts, but by MRI confirmation and post later on, these were confirmed as acute myocarditis. We used also a group of patients called, we call idiopathic DCM. So these are patients that have dilated cardiomyopathy, which have some signs of scarring in the MRI, and no history, no familial history of DCM. The other group is STEMI patients, because we were focusing on this difference between symptoms that are very similar, but a very different pathogenesis. And these were collected at the time, again, of presentation, like the acute myocarditis. And finally, we used a group of healthy controls, which were age and sex matched with the acute myocarditis group. So you can see here, we tested the presence of these c-Met T cells in the blood. You can see the beautiful cells here in this big blob, and in acute myocarditis and DCM, and you can't see them in either STEMI or patients or healthy controls. And these are diagrams with a number of patients that we have analyzed. For the benefit of clinicians, we looked at the sensitivity and specificity of this test, of the presence of these T cells in the blood, and compared with troponin T measurement, and you can see how much more sensible and sensitive and specific this measurement is compared to troponin when we compare STEMI patients with acute myocarditis patients. Now, these cells that we detected are in the blood, but are they also in the heart of acute myocarditis patient? And these are post-mortem, they're not biopsies, samples that we obtained from Cristina Basso in Padua. And you can see here, we stained for T cell marker CD3 and the c-Met. You can see quite a lot of cells that express both markers in the infiltrate of acute myocarditis. So they're not just in the blood, they're also in the heart. And then so what we did at that point is to go on and try to identify these T cells, because T cells are busybody. They do a lot of different things. And based on what they do, they define inflammation, adaptive inflammation in the body. So we looked at simply population of these cells. Again, this is in peripheral blood. And you can see we have naive T cells, which are the ones that are not being activated, central memory that have particular characteristics of trafficking, as well as effector memory that tend to go in tissue, but some are long-lived, and the TEMRA cells, which are exhausted T cells because they've been stimulated for quite a long time. And we really didn't see very big differences, maybe with the exception of the effector memory, between the c-Met positive and c-Met negative T cells in these acute myocarditis patients that we analyzed at this stage. What we then looked at were the markers of activation. So these markers tell you when a T cells has been activated. CD69, which is shown here, is an early marker of activation. So these cells have been stimulated in the body, likely in the heart draining lymph node, at maximum 48 hours before we did this assay. And you can see that the c-Met T cells subset, both CD4 and CD8, contain a lot more of recently activated T cells compared to the c-Met negative subsets. On the other hand, every type of immune response has an effector branch, but also has a regulatory branch, which makes sure that the immune response is not too exuberant and doesn't really generate bystander damage to tissue. And this is done by this subset of T cells, which are called regulatory T cells, or Tregs. And when we looked at c-Met positive and negative Tregs, we found that they were increased in the individual, in the c-Met positive subset. And they also, compared to the c-Met negative regulatory T cells, they also expressed a lot more of this GARP molecule, which again is an activation molecule that shows the potential of a Treg to suppress other immune response and maintain homeostasis. So this was quite interesting because it wasn't particularly novel in terms of what an immune response looks like, including an autoimmune response. What was incredibly exciting was when we looked at the cytokine expression profile. So cytokines are produced by the T cells, and they have different effect on both the T cells and other immune cells, as well as parenchymal cells. And we were surprised to find that the c-Met, which is subset that is the red here, and also in previous one, despite the fact that myocarditis is supposed to be a viral, initiated by a viral infection, they had a lot less gamma interferon producing cells compared to the c-Met negative cells. And this is quite interesting because normally antiviral T cells express a lot more interferon gamma than those that do other things. The other interesting observation was that these c-Met positive cells express IL-4, which is a type 2 cytokine, which was also associated with, normally Th2 responses are associated with allergic reactions as well as helminth infestations. These cells express quite a lot of IL-4, but also an effector cytokine that is called IL-17, which again is very strongly associated with autoimmunity. And finally, these cytokines called IL-22, which it has different effects depending on the milieu of the tissue inflammation that it is produced. We also identified within this c-Met positive cells two subsets are again very unusual. One that produced both IL-17 and IL-22, which was enriched in the c-Met positive population, and one that has only been described once in sites of implantation of the embryo in the womb, that is triple producer of these particular cytokines. So the importance of IL-2 I wanted to highlight because one characteristic, especially of the human heart, is that it is surrounded by fat, and IL-22 has been associated with lipid antigen presentation. So this is something that we are exploring at the moment, but I can't tell you exactly what it means. But just to say that this is a very unusual phenotype of autoimmune cells. The big question was really, are these autoimmune? These c-Met cells, are they autoimmune? And the answer again is yes, because when we looked at proliferation, so proliferation here is indicated by dilution of this fluorescence intravital dye, which is called CFSE. And when we looked at the c-Met positive T cells, they didn't respond to a nominal recall antigen tetanus toxoid, but they proliferated strongly against the autoantigen cardiac myosin. Vice versa, the c-Met negative cells proliferated strongly against tetanus, but did not proliferate much against cardiac myosin. So this suggests that really the majority of autoreactive T cells belong to this c-Met positive subset. And this is a summary of a number of patients that we analyzed. So now I want to, if you recall from the beginning, I showed you that the c-Met cells are also increased in idiopathic DCM. So again, we looked at their presence in the blood. We looked at their presence in the heart of these patients. And here you can see again that you have quite a lot of c-Met positive T cells in the heart of a DCM patient. Again, this is autoptic. And we did it on 5 patients. Now, what was interesting was that if you compare the number of c-Met cells versus c-Met negative cells in this DCM patient, it is skewed very strongly towards the c-Met positive cells. However, in total, the total T cell infiltrate, so the total CD3, are actually more in acute myocarditis compared to DCM. So when we looked at the c-Met positive cells and compared them with acute myocarditis and DCM, we found that there were a lot more c-Met positive skewed compared to acute myocarditis, suggesting that this population is the one that remains active during the chronic phase of acute myocarditis. So it really is selected during the chronic phase of the disease. Now, this is a part that I did, we did no, actually not yet. We looked at cytokines and everything also in IDCM, and the T cells, the c-Met cells there were very similar in terms of differentiation and production of cytokines, etc. And they were also specific for cardiac myosin, particularly the heavy chain. But there were some differences that were very telling. So when we look at the Treg cells, c-Met positive Treg cells, and compare them in acute myocarditis and DCM, you can see here that they are increased in high DCM compared to c-Met negative Tregs, which is what we saw in acute myocarditis. However, in contrast to acute myocarditis, the regulatory T cells in DCM patients did not upregulate this marker of Treg activity. So it looks like there is a defect in the c-Met Tregs of DCM patient. It might be related to the loss of tolerance and continuation of autoimmunity. So this is the part that we did in close collaboration with Perry and Alex. And we looked at familiar heart muscle diseases. So this was just something that came up of a lot of discussions. And I apologize, you know, this is when I do this talk to the immunologist, I have to explain cardiology, and when I give the talk to the cardiologist, I have to explain immunology. So I'm sure you will forgive me if I say stuff that you know already very well. So these familiar heart muscle diseases are caused often by mutations of genes that encode for structural cardiomyocyte proteins. They can lead to dilated cardiomyopathy. They have a very interesting, or at least some of them, occurs in flares, which are common perhaps common after infections. And there is a lot of evidence out there that describe the presence of inflammatory mediators in the blood of these patients, and signs that inflammation are present in CMR. They can benefit from some kind of immunotherapy. And so the question was, can we detect these c-Met cells in familiar heart muscle disease? And do they look like those that we find in AM and IDCM? And the answer is yes and no, because when we looked at these patients, and I don't have the table about but I think that there were a mixture of various mutations, and they were not at the stage of heart failure, that the ejection fraction was still preserved. What we found was that in these patients in the blood, we could only find the c-Met positive CD4 memory cells, but not the CD8. And this suggests that there is a different type of immune response going on in these patients. But also, and this is good news, we looked at these stem memory T cells, which is a subset of T cells which have stem-like properties, so it can renew indefinitely, and it's been involved in autoimmunity, the persistent of autoimmunity, the lifelong persistent of autoimmunity. And we found these T cells in IDCM, but we did not find them in the familiar familiar HMD patients. So to summarize this part, and I'm going to be very fast after this, I hope I've convinced you that circulating c-Met T cells can signal cardiac inflammation, and they're also present in the myocardium, so they're representative of the inflammatory process. They are enriched in autoreactive T cells. They're characterized by a very unique cytokine signature that might be important for the treatment of these patients. And they can display unique features in different heart muscle inflammatory diseases. So in part 2, I just want to show you some mouse data to tell you that these cells are actually causative of autoimmunity in cardiac inflammation, and that they might represent a prognostic marker for inflammation. So very quickly, we set up a model of experimental autoimmune myocarditis, which is induced by, is very similar to reproduces a human acute myocarditis in a number of aspects. and it involves 2 injections with autoantigen to break tolerance. And then a number of weeks until 28 days, which is normally the time where acute myocarditis happens in a number of mice, because this does not have 100% penetrance. It's only about 60%, sometimes even less. But not only did this, in these experiments, we blocked c-Met in a therapeutic manner after the second injection. So after tolerance has been broken in these mice with a c-Met inhibitor. This is a molecule that is a prototype of crizotinib, which is a c-Met small molecule inhibitor produced by Pfizer. And we did it not for a long time, but just a few days, because this is the time when T cells are meant to really fully express the homing receptor that are going to drive them to the heart. So the first observation was quite incredible. That is that obviously this is mice, control mice were just injected with the adjuvant without the antigen. This is the penetrance of the disease in our hands. When you treat the mice after tolerance breakage with a c-Met inhibitor, the incidence goes down more than 50%. So it's really, really, and I say incidence, but some of these mice would have developed myocarditis without the treatment in a therapeutic schedule. So what happens when we looked at the end of the experiment, we find that by looking at the number of mice, we find that the infiltration of lymphocytes in the heart is a lot less in the mice that have been treated with the c-Met inhibitor. And so is collagen deposition. This is a muscle trichrome staining of the experimental and acute myocarditis and those treated with a c-Met inhibitor. So just, I just wanted to show you this because in the mouse, and I'm convinced in the humans, these autoreactive T cells are the cause of myocarditis. Autoimmune myocarditis, and also we can block them by blocking this this particular receptor, this c-Met receptor. They're very similar. We did a full analysis. They're very similar to the c-Met T cells that we find in humans. And now I just want to give you the last piece of data, which is, can we use these T cells as a biomarker of cardiac inflammation, not just for the diagnosis, but also the prognosis of acute cardiac inflammation. So what we did was to look at the presence of the c-Met T cells in the blood of mice over the development of myocarditis. And you can see here, this is a control. There is a little bit of movement, probably due to a systemic inflammation due to the adjuvant, but you can clearly see that these cells really increase in the blood of the animals over time. And this increase is blunted if we block c-Met. What is important in this is that when we looked at these changes over time in mice that we didn't sacrifice at day 28, because in these mice, acute myocarditis can become chronic and lead to DCM after a long time for mice, which is 68 days. So we looked at mice, the presence of c-Met T cells in the blood and the histology, the feature of the heart of these mice. You can see that basically this is the majority of mice, have after, this is the control with adjuvant alone, after injection of the autoantigen, they have a little bit of extra c-Met T cells in their circulation, and then they go down. And these mice, at the end of the experiment, do not have any effect on the architecture of the heart. Some mice develop acute myocarditis and recover from that because the c-Met cells decrease after 6 weeks from immunization. And the heart in these mice is damaged a little bit, but not too much. You can see the damage increasing in some of the mice when they recover late, and this is accompanied by a late decrease of the c-Met cells in the blood. The damage is more, but it's not as bad as those that never completely remove the c-Met cells, lose the c-Met cells from their circulation, and these end up developing chronic myocarditis. So in summary of this part, I just wanted to say these c-Met cells are causative, certainly in the mouse model of autoimmune myocarditis, which is eerily similar to what happens in humans, that c-Met inhibition is therapeutic in this model, and that circulating c-Met cells not only can signal, but also predict the development and the transition to chronic inflammation in myocarditis. So I would like to propose that these cells might be a very good tool to address at least some of the issues that I presented to you at the beginning of this talk in myocarditis. And just want to finish to say thank you to all of these people. And I didn't mention the reason why John is here is that he didn't do the myocarditis experiment or it wasn't involved directly in this, but he gave me a very good idea, which is to look at whether these cells might actually be induced in the during lung infection rather than in heart damage. So it's a very important question, and we have some evidence of that, and I think it's going to be again another look at physiology to understand pathology. So thank you, John. Here are the people that did the work, Sylvia and Ed, both supported by the British Heart Foundation. I'd like also to acknowledge the help of Sam Mohideen, who's a consultant cardiologist, Professor Sam Mohideen, consultant cardiologist at Barts, who was really involved from the beginning and was overseeing the patient's sample and diagnosis and so on and so forth. I mentioned the collaboration with Alex and Perry, and I have to also thank all the other people that helped us with the, especially with the tissue, with the heart tissue and the analysis. And last but not least, the Barts Charity and the British Heart Foundation that funded most of the work that I presented tonight. So thank you for your attention. I have no idea what time it is. I think I might have gone over time, but I always do that and nobody stopped me. So your responsibility, Alex, Perry, and John.
Thanks, Federica. I'll hand over to John to start the questioning in a second. But Alex, do you want to just give us the rules and about asking questions?
Yeah, thanks, Federica. So essentially, we have 15 minutes for discussion and maybe a little bit more. So whoever wants to ask a question, you can post this in the Q&A section or in the chat, or raise your hand and we can unmute your microphone.
I'm just trying to stop sharing.
Great, thanks. So John, you're on mute.
Yeah, sorry. Perry, can I make a comment before we start the questions?
Of course.
I said at the beginning, what is needed is in science is creative idea of quality. I didn't add, and I should have, that what's also needed is courage. And I think in Federica, we have a scientist who has courage to say things that are a bit audacious. And she's prepared to take the flak from peer reviews about it. And I think we saw some of that courage of saying completely new things during this presentation.
Absolutely. So John, would you mind if I start off with the first question?
No, of course, yeah.
So Federica, thank you, tour de force. And I think it gives us a little window on the importance of defining the immune response in different clinical settings. I mean, as cardiologists, if we think about inflammation at all, I don't think we think about these different immunotypes that we see in the acute and chronic situation. But can I, just picking a few things out from your presentation, so loss of tolerance is clearly a key factor in the development of inflammation, but you showed us this loss of tolerance seems to vary between acute myocarditis, idiopathic DCM, and familial heart muscle diseases. So can you just say a little bit about that mechanism of loss of self-tolerance into autoimmunity and whether it persists or resolves? How does that work?
So this is $1 million question. Thank you. Only an immunologist could have asked that question. It's a big question. One of the weird things about the heart and its antigens is that the way the T cells are removed from the repertoire, if they are autoreactive in the thymus, is that the thymus has some transcription factors that allow the expression by the local antigen presenting cells of bits of peptides that belong to a number of autoantigens, including, I don't know, skin antigens. But there hasn't been the description of any cardiac antigen so far. And we're talking about quite a lot of peptides. So I don't know whether this has to do with the fact that maybe the heart and the thymus are so close, whether it's an anatomical reason or not, but it seems to be very easy to break tolerance against the heart compared to other organs. And it might be that it is broken much more often than we believe. We don't know that. Of course, that is a very good question. Regarding the progression, I mean, autoimmunity happens in a lot of diseases. So when you have thyroiditis, for example, you can develop anti-thyroglobulin antibodies. But then once you recover, that's it. They don't come back. And that is because even when you have autoreactive T cells in your periphery, there are mechanisms like the Tregs which can block that. And we saw that in IDCM, there seemed to be a defect in these Tregs. So they don't seem to become as activated as in acute myocarditis. So that could be a reason. There are other reasons, that, for example, as we discuss very often in familiar heart muscle disease, you might have a kind of chronic damage of the myocardium itself that create the danger signal that reactivate the autoimmune response. We need to define it. I think one of the things that I would like to highlight of what you just said is that we need to stop using, if we want to, if you cardiologists want to use immunotherapy in patients that have heart inflammation, you first have to understand the immune response that is happening there. So the majority of trials are very focused on innate immunity, and that is fine, but only if innate immunity is being the cause of the disease. In myocarditis, at the stage when the patient presents to the clinic, the innate immunity is already finished. It goes on because it's a vicious circle. So you need to understand what are you dealing with in terms of autoimmune disease and tolerance, breaking tolerance.
Thank you very much, Federica. Thank you. I'll move it on because we have limited time. Now, and I've got 2 or 3 questions, if I may. And Federica, you can answer yes or no answers if it's appropriate. You described DCM being related to gene mutation or gene change of some sort, often families. And then you have the c-Met causality that you've outlined. What is the relationship, do you think, between the gene change and the c-Met activation and its cause in macrophage activation and heart destruction? So
again, this is a very good question. The answer is what I was precisely discussing with Perry, that maybe the familial cardiomyopathies have the ability to generate stress in the myocardium, and the immune system will sense the stress in the myocardium and mount an immune response. So the gene would not cause the disease per se, don't tell the geneticist, but it might induce inflammation indirectly by making the cardiomyocytes suffer.
There's no evidence that there are genetically determined subsets of c-Mets, are there?
Not that I'm aware of.
No.
I mean, the c-Met knockout mice are lethal. So you have to, it's a very important cytokine for receptor for many other.
Yeah. Okay. I was convinced by your evidence that c-Mets in the blood of your mice were related to activity of disease. Now, one of the problems we have in clinical trials is selecting the population to receive a therapeutic agent. And as we know, many massive clinical trials have completely failed because the trial population is heterogeneous. And if you define a disease as a specific molecular or cellular change, you've got a whole mixture and you can never get a positive answer. Do you think that CMETs could be markers to select patients which would be responsive to specific anti-inflammatory treatment?
I mean, that is my hope, John, is my dream. If we can use it in such a way, then yes, absolutely. We can select those patients that, and you know, the point you raise, I think, is incredibly important in general for clinical trials. You know, that I just had to do with somebody for that is an expert in clinical trials, and all I hear is that we need to have millions of patients to find an answer. The reality is, if we select the patients that are going to respond or that are likely to respond, then we save a lot of money and a lot of headaches. So perhaps we should think about clinical trial being designed with the causality and the pathogenesis of the disease in mind, because, you know, for example, in heart failure, the colleagues tell me, in heart failure, you just put together HFpEF, HFrEF, whatever stage they're in. You're never going to have an answer. You're always going to need, you know, really huge numbers of patients.
Yeah.
And so
good answer. You spoke about acute myocarditis, but in dilated cardiomyopathy, DCM, you suggested that there was a causality of inflammation there involving CMETs. Unlike Perry, I'm a sort of amateur cardiologist, but my understanding is that DCM could be still a heterogeneous group of diseases. And could you not define a subset of DCM biologically by using CMETs?
John, you're a genius. I always say that. That is precisely what Perry and I are working on at the moment in a grant application.
Okay. All right. Very good. And my third question is, you didn't say much about ischemic cardiomyopathy. And traditionally, there is no immunological element in it. I just wondered whether ischemic cardiomyopathy implies breaking of membranes and ischemia and hypoxia and damage to tissue. Do you think there might just be an element of autoimmune disease in the myocardial damage of ischemic cardiomyopathy?
So I have currently a very talented CRTF, Dan Hardy, and he's looking at this particular aspect, because we looked at STEMI patient, and there we didn't find CMET cells, and we were quite happy not to find them, because coronary artery disease, by definition, is a vessel disease. It's not heart disease until you have the damage. There is the classical Dressler's cardiomyopathy, I think, after myocardial infarction, which is kind of late, and that is autoimmune. Whether we have a chronic inflammatory process that continues in some patients, we have to investigate. I mean, really, if you look at the breadth of cardiomyopathies that might have an inflammatory component, we have to rewrite cardiology from the beginning. You, me, and Perry.
Perry, what do you think, Perry?
Yeah, so I think there is some evidence that other aspects of the immune system are important in the remodeling process that takes place after myocardial infarction. So you have the acute response, but it's probably more about what happens in the chronic phase of remodeling, which is important in following anterior myocardial infarction, for example. I wanted to, so Federica, you were talking about the potential triggers, and at least on the data that you have in acute myocarditis, you were saying that you haven't seen a classical viral response. And this has been one of those, this has been a debate in dilated cardiomyopathy land for as long as I've been doing this, for 30 years. But you did seem to point towards an allergic signal, and also this potential. I didn't quite understand the the third component, which I think you were suggesting relates to lipid antigen presentation. I mean, maybe you'd want to just talk about perhaps the allergic component. And before you do that, it's condition that with the odd presentation that we see with acute form of myocarditis. So we get a young, healthy person who pitches up with intense inflammation, a heart that's not working. And if you support them, their circulation, sometimes that gets better. So what is it that's triggered such an intense inflammatory response, which happens and then goes away? Is that an allergic response?
Oh, gosh, this is tougher than my site visit. So the IL-4 component, I'm not going to say about allergy. I don't know whether there is any. Certainly there is a link with respiratory infections, but with allergies a lot less clear. But IL-4 is a major support for eosinophils, and eosinophils have been implicated in myocarditis and many types of myocarditis. So there might be a very important effect of these cytokines on the type of infiltrate, even before we look at it, once the disease is fully developed. I don't know. It's a very good question, but the data are the data. I mean, we find it both in human and in mice. It's not new. It's not something that we discovered. I mean, there's lots of data from Rose lab and that show precisely that. They're very strange cells. We do not know. It's a very fascinating disease from an immunological perspective, because so many things are different than a classic autoimmunity. The lipid antigen presentation, I think, is very exciting because, because I think it's exciting, but I'm an immunologist, so it might be just because of that. I think it's important because of the contact between the fat and the myocardium in humans. Is so, they really are close, and RVC, that it's invaded by the fat.
And it's also, as you've heard me say before, we see recurrent patterns all the time in clinical cardiology. We have, we don't understand what they mean, but one of those is the distribution of inflammation in the heart in acute myocarditis, and it's usually subepicardial, or often subepicardial. So what is it about that interface between the myocardium and the epicardial fat? And you're sort of giving us a tantalizing glimpse as to what may be happening at that interface to generate inflammation, I think.
Yeah. Can I just say something? I don't know whether there is any audience apart from you two and I.
There is. We've had a good audience.
If there is anybody that is enthused by study, we have lots of projects that we are trying to get going with Perry, with John, with other people at Barts. Cardioimmunology is becoming such an important discipline. So if you're interested, get in touch with me.
Okay. So I've got two quick questions. I was a bit disappointed that you left out the effect of the T regulatory lymphocyte on controlling the c-Met.
I didn't leave it out. So we showed that they are defective in the DCM compared to acute myocarditis. So that could be a reason why the DCM doesn't work very well. Sorry, they don't recover very well compared to the acute myocarditis. So there are differences in the regulatory. I know you're interested in regulatory T cells, John, but that is an important part that we're going to look at the transcriptomic of these cells soon. And hopefully that will tell us a lot more about the dynamics of these cells and how they talk to each other. But certainly the numbers are normal in DCM. They increase like they increase in any type of inflammation in the blood, and then they go to the tissue and they decrease with a different speed, as you know, depending on whether you give them a kick or not. But they seem to be less good at suppressing.
Okay. Okay, I think that's very important. Yeah. Okay, my final question, you mentioned the word atherosclerosis en passant. The beginning of atherosclerosis, I believe, is a macrophage in the intima of the artery doing something nasty. Do you think that c-MET positive cells might be in any way involved in the genesis of atherosclerosis?
So in terms of the phenotype of T cells in vessels, there are a lot of data out there, so I don't like a lot of competition. So that's why I'm focusing on the heart, and the vessels, I just ignore them for the time being. There is certainly a phenotype of homing to the vessels, but I don't think for example, fractal K receptor is typical of vessels that infiltrate the T cells that infiltrate the vessels. The problem is that there's still debate as to where the T cells that you find in the atherosclerotic plaque come from. So do they come from resident T cells that are sitting in the aorta or the artery, and then they move to the site of where the macrophages made the mess and there is a lot of fat? Do they come from the vasa vasorum, or do they come directly from the blood? So that is a little bit of a conundrum. And I haven't looked at c-MET expression in these cells, but my sense is that because we didn't find them in STEMI patients which should have atherosclerosis, then there might not be a case for c-MET in the vascular inflammation.
Okay, thank you very much indeed. It's after 5:30 now, Perry. So as chairman, I will close the scientific element of this before I ask Perry to close the meeting. But thank you again, Federica, for a really very stimulating talk, very courageous, and full, as I said in the beginning, of Italian Renaissance flair. Thank you very much indeed.
Thank you. I'll put it in my diary. This new definition is lovely. Thank you, John.
Yeah, thank you both. John, I've always seen you as a Renaissance man, and we've now got a Renaissance woman working together. So I think, Federica, thank you. As said, tour de force. And I think you said it earlier that what we're seeing, I think, is it's not a renaissance, it's actually the birth of a new subspecialty, I think, within cardiovascular medicine. We've tinkered around with a very primitive understanding of immunology and the treatment of immune disease in cardiology for too long. And I think now with the tools that we have, with the kind of knowledge that you're generating, and in a landscape where probably most of the therapeutics have already been discovered, what we're now entering is this new era, I think, of phenotyping, immunophenotyping, and trying to understand the role of the immune system in different cardiovascular presentations. And I think the future looks really exciting. So thank you, Federica. Thank you, John, for your masterful chairmanship, and thank you for the audience for sticking with us. See you next month.
Thank you. Bye-bye.
Bye-bye. Bye-bye.